Okay, so it's 1:02 and we've got quite a few people signed in, so I'm going to get going with this presentation. Thank you all for coming. My name is Joe Fredette. I'm Marketing Manager here at LGC Genomics, based in Beverly, Massachusetts, just north of Boston. The topic of today's webinar is our Zebrafish Genotyping Library, which we've just introduced, developed in collaboration with the Sanger Institute in the UK, Wellcome Trust Sanger Institute. And it utilizes our own CASP genotyping chemistry. So what I thought I would do is first give you a quick overview of the CASP genotyping chemistry and then tell you a little bit more about the library and how you can access it and information that's available on it. Let me switch to the slide view mode. Hopefully that's a little better view for you. So just a little bit about CASP. It stands for Competitive Allele Specific PCR, and for those of you wondering, it's not that we can't spell. Competitive is spelled with a K because it stems from KBioscience, which was the originator of the chemistry, the inventor of the chemistry. And about a year and a half ago, LGC Genomics acquired KBioscience. So the name sticks, CASP genotyping assay. It's a homogeneous fluorescence assay. There are no separation steps. It's PCR-based, singleplex PCR. We are coming out with a duplex version shortly. It's typically run in 96, 384, 1536 well plates. And it delivers accurate SNP and indel genotyping data and has a lot of flexibility, and it's also very cost-effective and requires very minimal DNA, so you don't have to do any kind of whole genome amplification to do your genotyping. And so, come on, page down. So here's just a quick overview of the components of the assay. The three components come together. You have your primer mix, which has your target-specific primers. And here we have two competing forward primers, one labeled in red here and one in blue. And then we have one common reverse primer. Importantly, these forward primers have these tail sequences that correspond to these fret cassettes, which are in our master mix, our universal master mix. So the master mix is the same for every CASP assay. The primer mix is different for every CASP assay, and it's target specific. But these tail sequences correspond to the fret cassettes that are in the master mix, and you'll see how that works as we go forward. And then the third component obviously is your sample DNA. And here we're showing a particular SNP location where it's either a G or a T in that location. So in the first round of PCR, you have your two forward primers that are competing. You do the denaturing of your sample DNA, and depending on what nucleotide is in that target region, whether it's a G or a T, one of the two forward primers is going to bind and elongate. They'll actually both bind, but only one will elongate, the one that has the correct nucleotide pairing at the SNP location. So in this case, the allele 1 primer binds with the C that matches with the G, and it will elongate. And then in PCR round 2, what happens is you end up denaturing, and then when your common reverse primer will bind to the amplicon you just created, which has this tail sequence with the allele 1 primer tail sequence. And as the common reverse primer elongates, it creates a complement to the allele 1 tail sequence, which is important for detection, as you'll see. Because as you go through successive rounds of PCR, you're going to create more and more of this amplicon with the allele 1 tail sequence and the complement to that allele 1 tail sequence, and you're eventually going to start pulling off the FAM-labeled sequence from the fret cassette in your master mix. It gets pulled off of its quencher, and it acts as a primer going forward. And because it's removed from its quencher, it's now fluorescing. So as the PCR reaction goes along through each succeeding cycle, you're going to see your fluorescence increase, and that's going to be an indicator of what allele the DNA has in that location. And if it's a heterozygous, obviously it will be a 50-50 mix of the FAM and hex. If it's a homozygous, it'll be all of one or the other. So that's just a basic overview of the CASP genotyping assay. I do have an animation here, and hopefully it works, and I'll just quickly talk you through it. It's basically what I just said, but just shows it in illustrative form. There's your DNA sample, and then you have your primer mix with its 3 components, the 2 competing forward primers and the 1 common reverse primer. And then you have your master mix, which is universal to all CASP assays, and it has the FAM and the hex labeled FRET cassettes that are quenched. So you start your first round of PCR, the DNA denatures. That's your SNP location for illustrative purposes. Again, it's either we're showing it to be a G in this location here. So the allele 2 primer comes down because it has an A at the very end, terminus. It can't elongate. The Taq polymerase comes down, but it won't elongate. When the allele 1 primer comes in, it has the correct nucleotide at the end, terminus with the C. Because it does, when the Taq polymerase comes down, it will elongate. So you're going to end up creating an amplicon with this allele 1 tail sequence. And then at the end of first round of PCR, you're going to denature, and you end up with an amplicon that has this tail sequence, and the common reverse primer generates the complement to that tail sequence in round 2 and in succeeding rounds. And then when you denature, eventually you're pulling off the FAM labeled portion of the FRET cassette, which has the same sequence as the tail sequence on the allele 1 primer. Here you're removing the fluorescence from its quencher, so now it's fluorescing, and as you go through succeeding rounds of PCR, that fluorescence is only going to increase. And again, if it's homozygous for one allele, you'll have all of one signal, either FAM or hex. If it's heterozygous, you'll end up with a 50-50 mixture of both. So hopefully that makes the CASP animation or the CASP chemistry easy to understand. One thing that's really nice about the CASP chemistry is our target-specific primers do not require any labeling, as opposed to probes, for example, in the TaqMan assay. Their target-specific probes require dual labeling, both the fluorescence as well as the quencher on the probes, and also the minor groove binding protein. Since those are target-specific reagents, they can't make them up in bulk. They have to make them up for every individual assay, which becomes expensive. For us, our labeling is all done on our FRET cassettes, which is in the master mix. So there is no labeling on our target-specific primers, and that confers a strong cost advantage, which translates into a highly competitive price. So this is just an overview of the CASP workflow. Again, the assay assembly is done in 96, 384, 1536 well plates. In our service lab, we do everything in 1536 well plates using water bath thermal cycling technology. The assays can go from 20 microliters down to 1 microliter. You got your 3 components, your DNA sample, your master mix, and your primer mix. You heat seal the plates. You do your thermal cycling reaction. When you seal the plates, you do it with a optically clear sealing film, so you don't have to ever remove that film. You just take that entire plate with the sealing film, put it into your fluorescence plate reader, or if you have a real-time PCR machine, you can actually read it directly on the PCR machine. And then you do your cluster analysis and SNP calling and data export. So that's the basic workflow. So a little bit about the Zebrafish library. We've been developing this in concert with the Wellcome Trust Sanger Institute. It's part of the Zebrafish Mutation Project. There's a link there for you to go to it, and we'll send this presentation out to everybody as a follow-up. We currently have over 1,000 validated SNP assays for known gene mutations, and they are spread pretty evenly throughout the genome across all 25 chromosomes. So very useful for mapping purposes, employing an out-cross, back-cross strategy. And this is really just the beginning. Working with the Wellcome Trust, they're looking to develop knockouts for every gene. And with every knockout, they're going to have a confirmatory CASP assay to confirm that knockout. Right now, all the assays we have are for known mutations, and they do confirm that mutation. And then we're just going to continue to build the library going forward. And I always like this picture. It always seemed to me like the fish were talking to each other, so I hope you'll indulge me here. Hey, Sally, any idea what he's talking about? Not a clue, Earl, but it sure sounds fishy to me. Anyway, this is the Zebrafish Mutation Project website. I'm going to take us to a link there so you can see what it is. They go through a process of mutagenesis, creating mutants, and then developing assays for those validated mutants. I'm going to click on here, and this will take us to the actual Zebrafish Mutation Project website. And you can see, they got markers across all 25 chromosomes, and you can search. You can look for mutant lines by either browsing the complete list or by searching for a particular gene. And you can request alleles and order up the zebrafish containing these mutants, and eventually the knockout zebrafish as well. And these are all sorted alphabetically currently. Again, you can search it by the complete list or by entering in gene name. If I just take a look at one randomly, say we'll take a look. Most of them have a normal phenotype. Some of them are associated with a unique phenotype. This is the kind of information you get. You get the ZFIN ID. You have the allele name. You can actually request the allele, get the zebrafish. And then if you look down here, you hear it says the CASP assay ID. That, by the way, CASP was an old name for CASP. We're going to get them to change that in the database. It is CASP going forward. And again, it came from KBioscience. It's now LGC Genomics. But there is the sequence, and that's the SNP location right there. And this is a validated assay for this particular mutation. So this is a very useful website to go poke around if you haven't already. And let me go back to presentation. And that's just the basic introduction, and I hope that you found that useful. Thank you for your time. If anybody has any questions, you can ask them now, or you can send me an email later, and I'd be happy to answer them. And when I end this webinar, there'll be a very short survey, 5 questions. It'll take you all about 3 minutes to answer. And if you would do that, as a thanks, we'll send you a free LGC Genomics mug. So thank you very much for your time. And with that, I'm going to see if I can get back to presentation here. See if there are any questions. Can we design new CASP assays ourselves for novel mutations? Absolutely, you can. We design new CASP assays every day in our service lab. It's a very quick process. In fact, we're very good at sometimes getting assays to work when they don't work on other platforms. We require a very small amount of consensus sequence to develop the primers for each assay, and all you need to do is provide us through our SNP submission form the sequence and the target that you're looking to develop an assay for. Incidentally, we can also develop assays for transgenes. The only thing about transgenes is you need to know exactly where the insert is. As long as you know where the insert is, we can develop an assay for transgenes as well. Thank you for that question. Okay, well, thank you very much. Again, when I end this webinar, you should see a screen that pops up with a couple questions. If you answer those questions and provide us with your address, we'll be happy to send you a mug. And thank you again.